Welcome to the Sand Science Experience podcast, where we explore the latest and most exciting papers and preprints in science. Before we dive into today's really fascinating findings, please do us a huge favor, like, subscribe, and share this deep dive with your friends and family. It truly supports the Sand Science Experience, and it lets us keep bringing you this kind of cutting-edge research analysis. Okay, so today we are cracking open a new paper published in Aging Cell. And this one, well, it really challenges how we often think about cellular longevity. The title kind of gives it away. Enhancing late life survival and mobility via mitohormesis by reducing mitochondrial calcium levels. Now, if you follow aging research, you know the usual advice is protect the mitochondria at all costs, right? This paper suggests maybe we need to stress them out just a little bit instead.
That's exactly right. It's a bit counterintuitive. So the mission for us today is really to get into how intentionally reducing mitochondrial calcium, that's Ca2+, somehow manages to extend not just lifespan, but health span too. The researchers looked at this in C. elegans, those little worms, and also in human cells. And we need to understand this mechanism they're calling mitohormesis. And, crucially, why the timing of this intervention seems so incredibly important.
Right. Let's start with the basics then. Why is calcium inside the mitochondria important in the first place? We usually hear about calcium for bones or muscle contraction. What's its job deep inside the mitochondrial matrix?
Oh, okay. Inside the mitochondria, its role is absolutely central. Think of it as a key metabolic regulator. It acts kind of like a throttle for energy production. Specifically, Ca2+ boosts the activity of key enzymes called dehydrogenases in the TCA cycle, the Krebs cycle. This basically ramps up the production of the fuel needed for the electron transport chain. It's like pressing the accelerator pedal for making ATP the cell's energy currency.
Okay, so it's a vital accelerator. And like any accelerator, I guess, you need the right balance. Not too much, not too little.
Precisely. That balance is delicate. The sources really highlight that. Too much calcium, or even too little, can seriously disrupt mitochondrial function. And what we see in age organisms, including the C. elegans worms they studied here, is that age-related decline is often linked to imbalanced mitochondrial calcium. Often it's actually increased basal levels. This kind of dysregulation is now being implicated in, well, a whole host of age-related diseases, neurodegeneration, heart disease, metabolic problems.
So a critical system goes out of whack with age, and the main gatekeeper controlling this calcium flow into the mitochondria is the MCU, right? The mitochondrial calcium uniphore.
Correct, yeah. The MCU complex is the highly selective channel. It sits on that inner mitochondrial membrane and carefully controls how much calcium gets into the matrix. And that's exactly what the researchers decided to target in this study.
Okay, so this brings us to the main experiment. The part that, as you said, feels counterintuitive. They used RNA interference RNAi to basically turn down the volume on the gene menu 1, which is the worm version of MCU. So they're deliberately reducing mitochondrial calcium. But if calcium is the accelerator, shouldn't that just cause a big energy crisis in the worms?
Yeah, that's the logical first thought. And, well, the results actually showed that initial hit. What's really fascinating, though, is what they call a biphasic result. It happened in two phases. So phase 1, yes, knocking down MCU1 did compromise survival early on in young adulthood. You're basically handicapping their metabolism initially. So there's this acute stress and, a temporary dip in survival.
Okay, a dip first, but then the survivors, this is where it gets weirdly good for them.
This is where it gets really interesting. The worms that got through that initial phase, they live longer. And not just a little bit longer. We're talking a significant extension in maximum lifespan, up to about 13.24% longer.
Wow, 13% is a pretty big deal in aging studies.
It is. And it wasn't just about living longer. They were living better in old age. The surviving worms showed really marked improvements in their motility. They measured how much they wriggled, basically body bends per minute. And this improvement was seen late in adulthood around day 21, which is typically when these worms show serious age-related decline.
So fitter for longer. That's the health span improvement you mentioned.
Exactly. Living longer and staying more active.
Now, you mentioned timing was critical. They had to do this knockdown before day 14. What's special about day 14 for a worm? Is that like middle age?
Yeah, you could think of it like that. Day 14 in an adult C. elegans is roughly the point where aging really starts to accelerate. It's like the onset of middle age and decline. It seems to be this critical window before too much age-related damage builds up. And the data was really clear on this. If they started the MCU1 knockdown after day 14, poof, the lifespan benefit was gone.
No effect at all.
None. Which tells us that whatever adaptive response is happening, it needs to be triggered earlier during this specific window.
Interesting. And they also found it was specific to certain tissues, didn't they?
They did, yeah. It had to be in the muscle. Muscle-specific MIC1 knockdown gave the benefit, but when they did it just in the intestine, it didn't work. So it seems linked to muscle health and metabolism specifically.
Okay, so inducing this mild early stress, specifically in muscle, before middle age, somehow makes them live longer and healthier. How? It doesn't sound very efficient, does it? It sounds more like controlled damage triggering a repair response.
That's a great way to put it, and that's the core idea behind Mitohormesis. Think of it like exercise for your cells, or maybe even like a vaccine. You introduce a small, manageable stress, and the organism ramps up its own defense and repair systems in response. And that boosted defense system then protects it better against the bigger chronic stresses that come with aging later on.
Okay, so what's the specific stress signal here, and what's the defense response?
Right, so the researchers mapped out this chain of events pretty carefully. First step, reducing mitochondrial C2 plus slows down the TCA cycle, like we discussed. That, in turn, makes the electron transport chain, the ETC, less efficient. They saw reduced activity in complex 1, complex 2, and complex 3.
So energy production takes a hit.
It does, temporarily. And this inefficiency causes some electrons to sort of leak out prematurely. This leakage results in a transient increase in reactive oxygen species, or ROS, specifically hydrogen peroxide, H2O2. And they saw this H2O2 spike happen right around day 14.
Ah, linking back to that critical time window.
Exactly. The timing lines up perfectly.
Okay, but wait, ROS. Hydrogen peroxide. For years, the message in popular science and even some research was that ROS are bad. Antioxidants are good. Fight free radicals. You're telling me this H2O2 speak is actually the good part, the necessary signal?
In this specific context of mitohormesis, yes, absolutely. This study directly challenges that simple ROS equal bad idea. They actually proved this ROS signal was essential. How? They gave the worms common antioxidants. Things like N-acetylcysteine NAC, or a mitochondria-targeted one called Mitotempo.
And what happened?
The longevity effect was completely wiped out, gone.
Wow, so if you mop up that transient ROS signal too early.
You block the beneficial adaptation. You need that little burst of stress to kickstart the response. Taking high-dose antioxidants all the time might actually prevent these beneficial stress responses.
That stands for, yeah, that's a big shift in thinking. Okay, so the H2O2 signal happens. How does the worm cell hear that signal and translate it into better defenses?
That H2O2 acts like an alarm bell. It activates a really important signaling cascade. This involves the worm versions, the orthologs, of key human longevity-associated genes. We're talking about PMK1, which is like our P38 MAPK, DAF16, which is the worm version of FOXO, and SCAN1, their NRF2.
Okay, these are known players in stress response and longevity pathways.
Exactly. They're master regulator genes. When they get switched on by that transient ROS signal, they basically tell the cell to beef up its internal antioxidant defenses. For instance, they saw upregulation of SOD2, a key mitochondrial antioxidant enzyme.
So the initial ROS stress triggers the cell to make more of its own antioxidants.
Precisely. It's an adaptive upgrade. And the end result of this whole cascade, it helps preserve the mitochondria themselves. They saw that this process mitigated the fragmentation, the breaking apart of mitochondria that normally happens with age. Instead, the mitochondria in the treated older worms stayed larger, more interconnected, basically healthier looking and functioning better.
How did they know they were functioning better?
They measured things like the NAD plus NADH ratio, which is a key indicator of metabolic health, and it was improved. And the basal oxygen consumption rate, basically how much oxygen the mitochondria are using at rest, was also better in late life.
So it's like they gave the cellular power grid a preventative overhaul.
That's a good analogy, yes.
Yeah.
Triggered by that carefully timed mild stress.
Okay, fantastic proof of concept in worms using genetics. But what about translation? Can we achieve this with a drug?
That was the next logical step, and yes, they tested that too. They used an existing FDA-approved drug called Mitoxantrone. Now, Mitoxantrone is primarily known as a chemotherapy drug, but it also happens to be known as an inhibitor of the MCU.
Ah, so it blocks that calcium channel pharmacologically.
Right. And they used it at a really, really low concentration in the worms, just 10 nanomolar. Tiny dose.
And did it work like the gene knockdown?
It did. Remarkably well, actually. It replicated that same biphasic lifespan response, the initial dip, then the extension. It improved motility in the old worms, and it helped maintain that healthier mitochondrial structure.
And the timing, did that still matter?
Absolutely critical. Just like the RNAi, the Mitoxantrone treatment only worked if it was given up until day 14. Start later, no benefit. So this really strengthened the case that Inhibiting the MCU, reducing that calcium influx, was the key mechanism.
Okay, that's promising. Worms are great, but what about human cells? Did they test it there?
They did. They used non-cancerous human foreskin fibroblasts, HFF1 cells, standard cell line for this kind of work.
Yeah.
They used a low dose of Mitoxantrone again, slightly higher at 100 nanomolar for the cells. And they saw the same pattern emerge.
What exactly did they see?
Well, first, it reduced mitochondrial calcium, as expected. Second, it inhibited the electron transport chain complexes, actually complex 1, 2, 3, and 4 in the human cells.
Yeah.
Third, and crucially, it induced that necessary transient spike in mitochondrial H2O2.
The stress signal again.
The stress signal again. And then, following that transient spike, about 24 to 48 hours later, they saw the adaptive response kick in. Enhanced expression and activity of key human antioxidant enzymes, SOD1, SOD2, and Catalase.
Wow. So it looks like the same mitohormetic mechanism works, at least in principle, in isolated human cells using a known drug. This sounds almost too good to be true. An existing drug, low dose, triggers a pro-longevity response. Why isn't everyone talking about this? What's the catch?
Ah, yes. There is a significant catch, a major caveat. The problem is Mitoxantrone isn't just an MCU inhibitor. That's not its main job, actually.
What else does it do? You mentioned chemotherapy.
Exactly. It's a potent topoisomerase 2 inhibitor, and it also intercalates into DNA. Basically, it messes with DNA structure and replication. That's how it kills cancer cells.
Okay, that sounds less good for healthy cells.
Much less good. Even at the low doses used in the proliferating HFF1 cells, these other activities, these off-target effects, cause significant problems. They saw acute cytotoxicity, meaning cell death, and sustained growth arrest in a large fraction of the treated cells.
So the drug does trigger the beneficial mitohormesis pathway, but it also causes serious collateral damage because it hits other targets too hard.
Precisely. It's like using a sledgehammer when you need a scalpel. The study brilliantly demonstrates the potential of targeting mitochondrial calcium to induce mitohormesis for longevity. But it simultaneously highlights why mitoxantrone itself is probably not the right drug for this purpose in humans. It's too dirty, pharmacologically speaking.
We need something much more specific.
Exactly. The field needs highly specific MCU inhibitors that only modulate calcium levels without those dangerous off-target effects on DNA and cell division.
Okay, so let's try and wrap this up. The big takeaway here seems to be that intentionally and carefully reducing mitochondrial calcium can trigger this beneficial stress response, this mitohormesis. It involves a transient spike in ROS, which then activates our own defense pathways, leading to longer lifespan, better late-life fitness, and healthier mitochondria in old age, at least in worms, and potentially applicable to human cells.
That sums it up perfectly. It's a really powerful demonstration of how a precisely timed mild stress can promote resilience and longevity. But it also leaves us with a really provocative challenge, doesn't it? If we think about translating this to humans, the implications are pretty complex. This research suggests the intervention needs two things that are frankly quite difficult to manage. First, it seems to require inducing a state that involves, at least temporarily, some reduced function or even cytotoxicity, that initial dip in survival they saw. And second, the timing is absolutely critical. It had to happen before middle age in the worms, before day 14.
Right. How do we even figure out what day 14 is for a human lifespan in terms of cellular receptiveness?
Exactly. And how do you ethically design and deploy a potential anti-aging therapy that by its very nature might make people feel worse or be slightly less healthy in the short term in order to achieve a potential long-term gain in health span and lifespan? It's a fundamental challenge for any therapy based on hormesis, getting that balance of stress and adaptation just right, safely in a diverse human population. That's the really tricky part.
A very profound thought to end on. It suggests the path to longer, healthier lives might not be about simply avoiding all stress, but about learning how to harness it precisely. Thank you for guiding us through that intricate dance between calcium, stress, and aging. That is all the time we have for this deep dive. Please remember to like and subscribe so you don't miss our next exploration into the latest science. This has been the Sand Science Experience.